Hello and welcome. My name is José Luis Ricón, and this is my talk about longevity. Here I've tried to condense not everything I know, but a lot of it into just 50 slides. Um, and even though it's not everything, you can read more in my blog here at intel.com/longevity. And you can also email me questions and stuff at my email address right there. Um, I'm going to be talking about, well, aging in general, what is aging, mortality and age-related diseases, also some conceptual issues with aging, like if aging is fixed or not in different species. I'll also discuss different subcomponents of the aging process, and then I'll go into different therapies that seem interesting. And then I will finish with some other comments about the robustness of the literature, how anti-aging drugs can get approved, and some controversies and open questions that remain, which are many. Well, this is, first of all, this is about, well, the biology of aging. This is not about the social consequences of aging or reversing aging. This is not about things like superpopulation or pensions or stuff like that. This is purely biology. Um, and also, this is not about like literally everything that that has been said to modulate aging. So this is not about whether exercise or diet can help or something like that. In this presentation, I assume as given based on health. So I assume that whatever the ideal diet and exercise and sleep are, whatever those patterns are the individual in question, they are already following them, so we can just strip them away. I of course have my own thoughts about sleep and about diet, but they are not the focus of this presentation. This is also not about immortality. So immortality is difficult to achieve. Like living forever is kind of difficult. So this presentation is not about that. It's not about whether or not we can live forever. It's more humble in its scope. It's merely about aging and extending life to some extent. And of course, well, I mean, at some point they kind of plan, but this is not about like radical life extension, so to speak. I think we are not there yet. Yes, so aging. So there are lots of debates about what aging might be. But I think a lot of these are merely semantic debates. People just like to argue about words. That's part of the nature of people. And so here instead, we're going to be just defining aging as in a very stupid way. It's just like you are young, you have more capabilities than when you are old. Everything from like the flexibility of your skin or your muscle mass, how your reaction to insulin, and glucose, um, and how competent your immune system is, and things like that. Well, of course, not everything that happens with age is bad. So here I give two examples. One is, well, as you get old, usually your knowledge increases, and you can argue that that's not part of aging. I don't care. I just think that that's something we will consider a positive change, and so I exclude it from my definition of aging. And also similarly, with aging, you also get some changes that are maybe responses to damage and things that are actually part of the aging process. So if something is a response to something we consider bad, then the response itself wouldn't also be necessarily aging in this regard. So basically, I'm mostly interested in something around the root causes of what actually degrades bodily function with age. That's more or less um, what I had in mind when I was making these slides. Now, here are some terms that some people use to talk about aging. The first one is negligible senescence, and this is the idea that so usually, and as we'll see in the next slide, mortality increases with age. So the older you are, the less likely is it that you're going to be alive the next year. Well, in some species, this is not true, and so they, in that sense, they don't seem to age. They seem to be, they still die, but they still seem to kind of, well, the mortality rate doesn't seem to increase. Then the second thing is slowing down aging. So this is the idea of taking an individual and then they will just age, but it's a slower rate. So here we are not reversing the process, we're not we're removing damage, we're just slowing down the process itself. Well, this doesn't lead to negligible senescence because well, mortality is still increasing even if it's increasing at a slower rate. And then lastly, we have reversing aging. This is the idea that you can take an aged individual and then bring it back to a more younger phenotype. and this could be used to achieve negligible senescence. So even if your therapy is incomplete and you reverse aging, but then you continue to age, if you continuously apply some therapy of this kind, which then you could actually, your mortality risk could stay constant, and you could stay more or less young for a while. Then there is the notion of aging versus agings. Aging, we may talk about the general process of getting old, so to speak. And agings, it may make sense to talk about the different processes that underlie aging. Aging sounds like a very vague thing, a very nebulous concept. And so if we want to talk about the specific causes of mortality or degradation of function, we may want to talk about aging of different parts of the body, which of course, and is one of the premises in neuroscience, is that many of these age-related changes have common causes. So we don't have to treat everything on its own. We can treat only a small subset of causes. As I promised, so here it is. This is how mortality increases in humans with age. So you can see that, well, after we are born, well, maybe people are born with some congenital disease or something, so maybe it's higher, but then it goes down. And then for a few years, the mortality risk stays constant, as if we didn't age at all, which is quite interesting. And then it increases again. It increases again, and it continues to increase exponentially until effectively you die. As a thought experiment, suppose that you could be, let's say, 10 years old or 15 years old forever, and you could have that mortality risk forever. So every year you're flipping a coin or you're rolling a dice and you have a 0.01% probability of dying. What will be your life expectancy? The answer is perhaps in the order of thousands of years, which is still, again, it's not in immortality, but it's quite a lot. This is not to imply that we are anywhere near this birth, but it's quite interesting to actually put a number into what could be achieved if we can just like revert you back to when you were that age. Now, with this increase in mortality, you also have an associated increase in so-called age-related diseases. Here I have at top left I have diabetes. To the right I have CVD, cardiovascular disease, and cancer. And to the bottom left, I have the various kinds of dementia. And in the case of, well, they all increase with age, and they all increase more or less in the same pattern. If anything, though, something that seems interesting is for the case of cancer, there seems to be a decrease in cancer rates after the age of around 80 or 84. And there are reasons for this, but as a general thing, I think that the generalization that cancer risk increases with age, I think it's still more or less valid. Well, I will discuss later why there are hypotheses about why this decrease in cancer risk might be, but I will not get into that right now. As I mentioned earlier, the basic idea in the longevity field is that we can take all these diseases and things that happen with aging, compress them into a smaller set of causes, and then devise an array of therapies that can be used to address all of this. And it can be that, for example, if you take arthritis, we may say, okay, arthritis happens because, let's say, of senescent cells, which is one of the things that's here, and perhaps also because of this thing called cross-links accumulation, which we'll discuss later. And maybe cancer is because, well, you accumulate mutations, of course, but also maybe your immune system gets weaker for reasons we'll discuss later, like invasion of the thymus. And so we map these diseases to these causes, and then we treat those causes individually. And so, for example, let's say if senescent cell accumulation causes arthritis and cancer, then a therapy to address senescent cells may have an effect on arthritis, cancer, and many other things. This is very much unlike the way that medicine is typically done. So if you take, for example, one of the most popular drugs, which is statins. Statins are typically used to lower LDL cholesterol to avoid heart disease. And in principle, that's what they're used for. It's not that you take them for general rejuvenation, reducing general your cancer, mortality risk, and other things. But in this approach, you're explicitly looking for these common causes that may affect a lot of things, to kind of go into the root causes instead of the proximal causes, going to the deeper causes of the disease. It may seem that living for a long time it's difficult, so you can say, well, there is entropy, damage happens, and we can cope with all of that. Well, maybe, but well, some animals clearly can live for a while more than us. The longest lived animal that I've been able to find is this thing called the ocean quago. It's like a huge big clam thing, and it lives to like around 400 years. Whales are mammals that also live to 200 something years. And well, there is a shark that lives to 400 or 500 years. That's not in here. Um, but you get the idea that animals there, it's not purely theoretical. There exist already animals are not just like animals very different from us, like I don't know, like worms or like plants, which are not even animals, but animals that are closer to us, like fish or mammals, that live longer than we do at their maximum lifespan. And this, of course, in the wild, but this is without any healthcare. Like you don't, like whales don't go to the doctor and get like surgery for heart disease or something. So you could imagine that maybe with better treatment, they would live even longer than they already do. Each of these animals, they have unique adaptations. So, for example, if you think of whales and elephants, which are quite big, and also elephants are long-lived, they have a lot of cells. Now, if you have a lot of cells, and given a constant probability of each cell becoming cancer, it may seem like the bigger animals should like they are at a high risk of dying from cancer. And so these animals have developed mechanisms to actually to prevent cancer from spreading. So in the case of elephants, for example, you have extra copies of a gene called P53, which in humans, maybe 50% of cancers or more have mutations that disable this gene. So they have like extra copies to make sure if there's cancer, this gene will act to stop it. Well, if and then, so to the bottom left, we have this picture of this weird looking thingy. It's called a naked mole rat, and it doesn't live that long. It lives for 30 years. But compared to animals that are similar to a naked mole rat, like mice, which live like maybe 3 years or so, it lives 10 times longer, which is quite a thing. So, and it also has its own adaptations. So I think one of the most famous one is one called high molecular weight hyaluronic acid, which it's a substance that exists in between the cells in the naked mole rat. And this substance seems to prevent cancer in reasons that are in ways that are being studied right now. And there is also one study, by the way, going on to say, okay, can we just take this thing and just put it in mice and see if they live longer? So there's a study that's coming out from the PI in the lab is Vera Gorbunova from Rochester University, and she's doing a study where they have transgenic mice that express the gene that generates this high molecular weight hyaluronan in mice. And so we may see mice that are extremely cancer resistant using this gene. And well, who knows? Maybe in the future we'll have gene therapies that leverage this specific adaptation. So as I say, part of the whole longevity stuff, well, in the same way that we stole CRISPR from bacteria and a lot of progress is coming there just from stealing things from nature, it may be that a lot of longevity may involve just going there and then just taking what's already being made for us in nature. You can take all those adaptations, and maybe they work, maybe they don't, but trying maybe helps. Even for a single species, so in this case, C. elegans, a worm, even for a single species, aging can be changed. So it's not fixed for a species. In this case, if you take like a random worm, so-called wild type, it lives for maybe a matter of weeks. In this case, it's like 25 days. But if you change just one mutation in just one little thing, and it lives 10 times longer. And these worms, they look otherwise healthy, so everything is fine. It's just there was like this gene that is doing something in the wild type, and it makes them live longer. So why might this be? Why is it that there is just like knock out this gene and then they live 10 times longer? Well, it may be that there was no reason for evolution to select against this gene. Maybe it doesn't do anything bad while the worm is within, let's say, 25 years of age. It only becomes a problem to have that gene or that particular set of nucleotides if you are older. So in humans, it may well be that because evolution doesn't really care about you after you have reproduced, it may be that we have genes that either are bad for us in an active way that stick around because they're not selected against, or that we have genes that are good for us when we are young, but when we get older, then they are bad for us, and they cause aging. And so again, this is also one way we'll discuss in a moment about this whole thing about genes. So here it is, this thing about genes. So in the case of humans, let's begin by looking at the first point. So there is this this is SNP, this SNP called RS2802292, which this specifies a specific position in the genome. It's a pair of letters. In this case, having GG in your DNA. So if you have this in your GG means in the two copies of your chromosomes. So if you have this this allele in this SNP in FOXO3, a gene which is related to various things in longevity, but I will discuss that here. You're twice as likely to live to the end 100, which is quite interesting. So this means that you can just find the set of these genes and then just like just change them, and then you get already some benefits from just from taking what we know, because there are already people alive that have these variations. So we can so we know they are not harmful, or in as much as they are harmful, it doesn't seem to be that much bad, because they still live a long time in a healthy state. And there are also other genes that are not directly aging related, but that also matter. So there is this gene called BRCA, or BRCA, that increases greatly the odds of having breast cancer. Now, you could argue that just breast cancer is not aging, but you can also think that if you want to reduce mortality risk, this is not the only one that you can target. So it's not just about general mechanisms for aging. You can also go and find all those genes that have large effects on mortality, and think of ways to modulate them. Another one of these ones is PCSK9. So this one, there's currently a set of therapies that try to target. So PCSK9 has a role in LDL cholesterol homeostasis, or rather regulating how much LDL cholesterol should there be in your blood. And if you have a mutation there, then you get more LDL, and more LDL means more atherosclerosis. Well, in this case, more LDL particle count, but atherosclerosis, and that's bad. Okay, so you could also fix PCSK9 in theory, and that will also get you more lifespan and less atherosclerosis. But you will still accumulate the atherosclerotic plaque, but at a slower rate, which again, it's something. So when looking at genes, we usually found that many things are heritable. So for example, famously for intelligence is highly heritable. So if your parents are smart, you will probably be a smart kid. But with lifespan, it doesn't seem to be quite so. It seems to be more like 10 or 30% heritable. So if your family is long-lived, you won't necessarily be long-lived. Or if it's a if it's a twin that if they're like two twins and one, so that their age is not hugely correlated with each other, showing that it's not just the genes, also the environment also explains some things about that. And well, not also like different access to healthcare, and also just like pure chance and randomness, perhaps. So, for example, if you take atherosclerosis, high LDL cholesterol will increase your odds of having heart attack, but you may never have one. Maybe something else will take you first, and maybe or maybe not. So who knows? So the moment if you just take genes, you can predict about 8% of lifespan just from the genes that you carry. So to the right of this thing, we have the survivor curves by polygenic risk scores for various cohorts. So the idea here is that you take it's usually a linear model, and then from this linear model and the genomes of various people, you give them a score, the polygenic risk score, and then you take the ones that score the highest in the thing and the lowest in the thing, and you compare them, and you can see that women live longer than men, as you might expect from aggregate studies, but also that the effect, these predictors are not that great yet. So if you look at both between men and women, you can see that the predicted, in this case, the maximum lifespan predicted is about the same. Well, sorry, these are not predicted; these are actuals. And also, the mean lifespan. So this when we take the difference in the curves at around 50%, maybe just about only just a few years. And so again, because of this reason, gene therapy matters. So it may pay off to actually invest and think about in ways in how we can safely and cheaply change the genes that we already have in order to either fix these genes that we already know that are problematic or can be beneficial, as in the case of the FOXO3 mutation, and also to possibly to add some other genes that may be beneficial in the future. So finally, the longest lived human, although this is disputed, some people argue that it was her daughter that took her place at some point. It was a young man who lived to 122. We may not believe that one, or we may, but in any case, we also have the example of Saragnaus, who lived to 119. These are like you can see this as how far you can go living as a human being. Most of the supercentenarians are usually women. I think the longest lived man right now, I think it's Spanish guy, I think it's 105 or so. Yeah. So now we get into the idea of aging frameworks. And by aging frameworks, I mean high-level ways of conceptualizing and thinking about the aging process. The most common one, if you go to read papers, the one you're going to find is so-called hallmarks of aging. And this model is about 7 years old. It's a very popular model in the field, and you will see many papers talking about the hallmarks. And so a hallmark is something that, according to the paper, meets three criteria. One is that it gets worse with aging. Two, if you reverse it, then you reverse aging. And three, if you make it worse, then aging, some sense of aging gets worse. So the idea here is that it's not necessarily that they are any kind of root cause of aging. They are explicitly not meant to be root causes. They are meant to be things that are somewhere in the causal chain that leads from aging or something to a degradation of function to some extent in some concrete way. These are not also the complete set of things that change with aging. It's just one particular conceptualization. So there, and even though yes, I think so. The best way to think about this is more like if you look at things that change, just take them and just put them together, and they are not necessarily all the same, and they try to group them into primary, antagonistic, and integrative, but they're not quite like that. So mitochondria, they also have dysfunction on their own. You also see loss of proteostasis. It also has its own causes. And also, of course, like cellular senescence can directly induce, we'll talk about that later, cancer and other things. So it's a model. I mean, it's not the model. It's one model. One can think about aging, but it's one that is very popular. So I have to at least mention it. Another model is the model from SENS. So SENS is a study from negligible senescence, and this comes from the SENS Research Foundation, headed by Aubrey de Grey, or founded by Aubrey de Grey, back maybe a decade ago or more. And so the idea here is that, like fixing the body is kind of hard, since we're going to focus on maintenance. We're going to say, okay, if you have like cholesterol, doesn't matter, just let the cholesterol build up, making into. And then we can just like, if there is this atherosclerotic plaque, you can just remove it. Or if there are senescent cells, it doesn't matter where they come from, we just remove them. Or I don't know, if you get cancer, we just cure cancer. There are like various things like that. So to recap, it's not identifying root cause of aging, but they are more coherent than perhaps on the hallmarks, because these ones are intended to be, they're designed in such a way that in theory, according to their framework, and this is controversial, very controversial, if you cure these things and only these things, then you can fully rejuvenate someone so they can live, again, as an example before, from the 20-year-old that lives for a very long time, or 10-year-old. Another good thing of SENS that I like is that not only they pinpoint things that there are damages that they should be repaired, but also they say how those things should be repaired, which again, you can disagree with them, but at least they have some kind of plan, which not that many people have. They have like a comprehensive plan. What they do not have, I think it's not to blame them because it's something that no one else has either. It's a roadmap to say, here's what we know, here's what we don't know, here are the different ways that this could be approached, here's our current knowledge, and here are the blockers to get to a proper therapy for this whole running zone. And also, well, historically, well, even though they have been, to their credit, the Sense guys have been making progress treating across some of these damage types, but they have been historically quite optimistic. So there, it's taking its time, although it's not that they have loads of funding anyway. Another one, this one that I came up with, trying to think about, okay, what are the root causes of aging? What are the things that actually are driving the things that are not driven by anything else? In some sense of not driven by anything else, because of course there is always a cause for anything. One of them, so the first block, I think it's information loss, which is a consequence of stochasticity, or rather a combination of two things. Stochasticity plus imperfect or not 100% perfect repair mechanisms. So DNA mutations. So suppose you have one place in your genome and it changes one letter. That a cell can maybe it can repair it. It can say like, hey, this has changed, or maybe rather than change, let's say one of the letters in your DNA has suffered some damage, that molecule that's there, like a cytosine. So then the cell can probably repair that one. And in fact, for example, if you look at not mutations, but things like DNA double-stranded breaks, so when you have like the DNA helix and then it breaks, there are literally hundreds of thousands of like these breaks happening in cells in each cell every day, and they all get repaired. So these mechanisms are very good, but they're not perfect. And so mutations can still escape from these mechanisms. Likewise, changes in the epigenome, so-called epigenetic noise, is something that has been pointed to as a driver of aging recently, and it's for the same reason. So we have enzymes like DNMTs and the 1011 translocases that maintain or upkeep the epigenome, keep it the way it should be. And we'll talk about the epigenome later, but the point here is that these things are kind of like the programs from which everything else is coordinated. And if this breaks down, then everything else that depends on that also breaks down. And also, we'll talk about, there are some potential therapies for epigenetic noise and for DNA mutations. We'll also talk about that. But there are also things that happen in our bodies that lead to some hallmarks of aging, or things that come with aging, that are meant to be so by design. So telomeres. Telomeres, again, we'll talk about later, but they shorten as we age. They are meant to shorten, so the way DNA replicates, they shorten. It's not some random damage. They are meant to shorten by the way it works. That shortening leads eventually to senescent cells. So senescent cells may be a damage you want to eliminate, but the cause of senescence is the telomere shortening, in most cases. We'll talk about it later. And also something else that may be of interest here is depletion of initial stock of cells, tissue, protein. So this is, I have in mind, for example, the involution of the thymus. So the thymus is this gland that we have around here in the chest that trains T cells. T cells are so called because they come from the thymus, T. And this thing shrinks as we age and becomes a lump of fat. The same thing happens with the adrenal glands that also secrete hormones. And there may be many other things like this. And so again, so the thymus involutes or shrinks is not because of damage. It's not because there is anything wrong with it. It happens in every single vertebrate there is. It's programmed in that sense. And there may be evolutionary reasons why that happens, which again may have to do with it's good to have some thymus when you're younger, and maybe when you're older, evolution thinks you're not going to need this. Let's just throw it away. But maybe if you want to live longer, maybe you need it. It's something like this, so you may want to keep it around. And lastly, we have the accumulation of things that your body cannot deal with, again, by design. So suppose one is the accumulation of intercellular junk. One example is lipofuscin, which is a bunch of a blob of like lipids and proteins and some metal ions. It's like bunched together. And although this is a matter of debate, but that the consensus seems to be that there is no way that the body can get rid of this. It's just like nothing in our biology that can get rid of lipofuscin. So if you have cells and this thing just accumulates inside of them, and it won't get out, It won't get degraded no matter what drug you give to the body. There are no enzymes to break it down. As I say, some people claim that there is, which maybe could be great, but this is kind of like the idea here, that things inside the cell that cannot be broken down accumulate. And lastly, we have things that accumulate in the same way, but outside of the cell that we cannot deal with. One maybe the most famous one is the accumulation of amyloid or amyloid fibers or plaque, which may or may not have a role in Alzheimer's. There was a recent debate about if it matters or not, or to what extent. Some people say it's the tau protein, which is a protein. But in any case, that would be one example. Another example would be advanced glycation end products, which we'll also talk about later, and atherosclerotic plaque, which again, when you get, in this case, it's not so much that the body cannot deal with it, it's that it cannot deal with it beyond some level. So let's say if you get like a small lipid and it sticks into your into the wall of the artery, then it gets some inflammation, and the immune system will try to clear it out, and maybe it succeeds, and then you're fine. But if there is too much of it, then you get this plaque, and then there is no way back. At least that your body cannot do anything about it. So this is why currently, for people that have high cholesterol or have lots of plaque, they just get statins to reduce the rate at which this accumulates. It's mostly thought to be irreversible. Well, you can, in some cases, you can actually open some arteries and then just take out the plaque manually and just close them back. But that's you cannot do that. You cannot do that in all of them, and it's not like a riskless procedure. In any case, so this is what I was thinking about what the root causes might be. Of course, there might be more root causes, and the fact that these are root causes doesn't mean that they should be targeted therapeutically. So, for example, it might well be that it's easier to just go after senescent cells instead of trying to do anything with telomeres. Or we may think that actually it doesn't, or it's going to be extremely difficult or more difficult to completely prevent or repair DNA mutations than curing cancer, which both things are extremely difficult, but maybe addressing cancer, it's easier than dealing with mutations. So mutations accumulate at a slow rate. So maybe if we fix everything else and we find something to cure cancer, maybe this is not that bad. So maybe we can buy some time to work on this problem. Okay, so now we jump into individual topics. So going a bit over individual things that are part of the aging process. The first one is one of the hallmarks of aging also, and it's called genomic instability. This is the idea that, well, the genome, as we age, there is more DNA damage for various reasons. There is also the activation of transposons, which are maybe a substantial, like double-digit percentage of our genome are these genes, these sequences that their only purpose in life is to copy themselves into other parts of our DNA. That's everything they do. They don't help to do anything. It's like the ultimate selfish gene. And these guys, so these guys are typically suppressed. So they are not doing their thing. But as we age, this suppression, which it's due to the next one that we'll discuss, which is damage to the genome, this expression generates lots of mRNA. So you have all these like messenger RNA being produced from the DNA that would that kind of makes it difficult for the cell to kind of actually, like the more of this mRNA you have, so the way you're making proteins is that mRNA has to collide or get to the ribosomes to be made into proteins. So you have all of this mRNA going on from random things. The right mRNA may not get in sufficient amounts to the ribosomes, and you don't get the right proteins. So it's kind of like introducing, just like increasing the noise in the whole process. Well, while we are young, cells can fix most of this, so it's not like we are completely at mercy of these changes. But these mechanisms are not perfect, and especially if the mechanisms themselves degrade, then, well, of course, they're not going to repair themselves. Genetic instability may be especially damaging in stem cells, because if you think about it, if you have a cell and it's very damaged, the cell could in principle just die, just undergo apoptosis and be removed, just like be eaten by macrophages. And then a stem cell just can divide, um, and then another cell can just take its place. So it doesn't seem that bad, but if the stem cell is damaged, then, well, if it's a mutation in the stem cell, then the child of the stem cell are going to inherit the mutation. And also if the stem cell is damaged, very damaged, then it won't actually replicate. So you're not going to be replenishing lost cells with those stem cells. So that all said, mutations in the DNA, well, they of course can lead to cancer, which is kind of bad. But other than that, it's unclear to what extent drive aging. So compared to other things, as in if we manage to fix, so suppose that we can just wave a wand and you can like fix all the DNA mutations and you never get cancer. That probably wouldn't do much because like everything else will probably take you down before DNA mutations become a problem. Although this is not to say they're not a problem, they are, but it will take a while. The same is true in mitochondria. So with mitochondria, the powerhouses of the cell, they have their own mitochondrial DNA, and that also gets damaged to the same effect. It also can cause issues for the cell in general because, well, the cell depends on this mitochondria for energy. So I talked about telomeres earlier, but I didn't say what they were. So telomeres are these in chromosomes, which is like bundles of DNA. The telomeres are like these end caps that are at the ends of the chromosomes, and they are basically they have this like structure that's like called a T loop at the end, protected by this bunch of proteins that are collectively called sheltering because they shelter the telomere. The point of the sheltering is that the telomere looks like so to the machinery that repairs DNA damage, the telomere looks like a huge DNA break. It's like the DNA is supposed to be like a continuous thing without breaks, but the telomere is just like ends there, so it's like a break. So if you take the sheltering out, then the cell just like dies. It seems like, oh, this is so damaged, we cannot keep going. But also conversely, the sheltering means that the telomeres are not going to be repaired. So if they have damage, that's it. They will get damaged. And so, well, for various reasons, be it damage, and also because when cells replicate, their telomeres shorter by design. As this happens, the cell enters a state of so-called cellular senescence, where they don't replicate anymore, and we'll also talk about that later. But it's not a great thing to be a senescent cell. There are also reasons why a cell may be a senescent cell, but probably telomere-induced senescence is probably the main one. And now, one potential use of telomeres, one reason why we have this mechanism in there is as a tumor suppressor. So if you're a cancer cell and you're replicating, well, your telomeres are shortening all the time, and at some point they are super short. So then this mechanism is going to kick in and stop the cancer from replicating, and that's great. But the problem is that sure, I mean, maybe it pays off as a whole to have a mechanism that if there's like 0.001% of cells that's going to become cancerous, it's going to have a mechanism to address those. But for the 99.99% of cells that are fine, but when their telomeres shorten, they are going to get senescent. They're going to get into that state, even though they are perfectly fine. They are not damaged. They are perfectly functional. It's just that their telomeres are short. So in this case, our biology is making a trade-off between suppressing cancer and perhaps and making cells senescent, which on their own also decreases lifespan. Now, telomeres are kind of controversial, well, to some extent. So if you remember from a few years ago, there were maybe like popular science books and things about like, oh, telomeres are so important, like, telomeres are the key to live longer and stuff. A lot of people said like, like telomeres, like if you have long telomeres, like your cancer is going to get out of control. You have some people like, think of like Brett Weinstein, like, oh, like mice have like long telomeres because we have bred them to have long telomeres, and it's kind of bad. Well, okay, let's dispel this myth. Telomere length does not predict lifespan across species. The telomere attrition rate is what predicts such thing. So, for example, in mice, you can see in the graph to the utmost right that telomeres in mice, they shorten at a rate of 7,000 base pairs per year, and in humans, as you can see, it's only 70. That's a huge difference. And if you can see, you can actually, if you try to plot telomere length and maximum lifespan, you don't really see a relation. But if you plot the rate of shortening and average lifespan, then you see a relation. So, for example, in the paper where this comes from the lab of Maria Blasco, they did this experiment. If a human had a telomere 75 or 50% of their length, how much would their lifespan be? It wouldn't be that much change. Like our lifespan wouldn't drop by half or anything like that, because they will still shrink by a small amount. Now, I said that telomeres work as a tumor suppressor. So intuitively, it would seem that short telomeres should inhibit cancer, and conversely, long telomeres should lead to not cancer. But actually, that's not necessarily true. So there is again this paper that from the Blasco lab, where they create mice with super long telomeres. Well, if these mice lived longer, they got less cancer, and they developed in general less age-related pathologies. Why might this be? Well, because short telomeres means that sure, you will get if you get cancer, the cancer will spread less. But for all of the other cells, they're going to reach senescence earlier, and senescent cells on their own can also lead to cancer and other diseases. So in the case of mice in particular, because they have such a high attrition rate, these super long telomeres work for them. So, and also I forgot to say, in humans, if you have a rare mutations that make your telomeres extremely short, then in that case your lifespan is going to be short. But for most people, at least in a clinical setting, it's unclear yet if telomere length does anything to predict lifespan. There are even some studies using Mendelian randomization. This is where they look at the effect of single genes in telomere length. And excluding these rare mutations, it seems that telomere length in humans is marginally increases the odds of getting cancer. So longer telomeres, slightly higher of getting cancer, but also slightly smaller odds of getting cardiovascular disease and other things. So they concluded that overall, your total lifespan is unchanged in this case if you have different length telomeres. I also want to use this to remark that the telomere thing and the whole thing about telomeres comes from, well, there was like this time back maybe decades ago where you had like different theories of aging and people arguing about like all these telomeres and this sort of thing, and they were like really all about this one thing. And now we know that there are many more factors implicated in aging. It's not just the telomeres, and also of course that the story wasn't as simple as all telomeres are cancer suppressors. It's pretty much context dependent and more complex than we initially thought. Okay, so now we have epigenetic alterations. So as a fresh recap, so the DNA in your cells it's almost the same. Like in T cells it's not quite the same, but mostly in most of your cells it's the same, but your cells are not the same. Like neurons are not the same things at fibroblasts. The reason they are not the same is the epigenome. So the epigenome is kind of like metadata that you have in your cells that tell the machinery in your cell about how to execute the DNA program. So as you can see, the DNA is coiled around these little balls called histones, like I think it's like a bunch of histones to form a so-called nucleosome. The histones have tails, and on these tails you can get different modifications such as methylation or acetylation, which are like when you get an acetyl or methyl group into a tail. And getting those groups in there will open or close down the structure of the DNA, or like this kind of like so-called the chromatin, which is this high-level compactification of DNA. Besides this histone business, you also have on the DNA itself, you also have so-called CpG sites. They're sites that are a C and a G, a cytosine and a guanine in the DNA, and on some of these sites you have a methyl group, and it's so-called. In that case, we say that site is methylated. Methylation, so if a site is methylated or not, will determine whether or not that the gene gets expressed. And so, for example, if you are a neuron, you want to suppress genes that will be useful only if you are a cell in the liver. There is a problem with this though, that the epigenome is very flexible. So the epigenomes of our bodies, they are meant to switch on and off different parts of the epigenome to react to their conditions and also um, to, uh, to define cellular identity. Um, so the epigenome is not as stable as the genome. And because of this, as we age, the epigenome gets damaged. So cells lose these methylation marks and they steadily lose their identity. Which means that that's not great because maybe you're going to get more expression of some proteins or less expression of some other proteins, and that's going to affect you across the board. Perhaps a few decades ago, this was not seen as obviously a driver of aging, but now I think it's becoming quite clear that this thing is quite key to the whole process. Fortunately, though, unlike with DNA mutations, there are some very promising ways of addressing the epigenetic alterations that I will get to discuss later in the section about therapies. Did I miss anything? No. Okay. The next one is loss of proteostasis. So proteostasis is the idea that in a given cell you have to have a given concentration of proteins, um, and also that if a protein gets misfolded or unfolded, because proteins are produced as a string of amino acids, and that they on their own are supposed to kind of like put themselves together. But if that fails, many things can happen. Ideally, you get these small proteins called ubiquitins attached to the unfolded protein, which then carries the unfolded protein to the proteasome to be degraded, and that's fine. You can also get these other proteins called chaperones to help the protein fold itself again, and then it's fine. You get a folded protein, or you can also again with the help of chaperones you can get these proteins into the lysosome for them to be broken down in a different way to the proteasome. However, if for some reason these mechanisms are broken down, and also purely because of stochasticity, like sometimes it's like proteins just like collide in some weird way, you get an aggregation of proteins that we don't have the means to break down, and then it accumulates. And again, as I mentioned before, some examples of these are amyloid tau aggregates or levofuscin. And also, it's not just accumulation of aggregates, it's also the under or overproduction of these. So for example, one example that I recently found is that as we age, some people, we seem to be deficient in zinc, in the metal zinc. And this is not just for dietary reasons. This is because the actual transporter proteins, the proteins that take zinc into or outside of the cell, are over or underexpressed, which means that the cell is not taking, so cells are not taking in a sufficient amount of zinc, which then can apparently lead to issues with the immune system. And also, well, it can also make it worse because zinc also plays a role in methylation, and methylation helps keep the epigenome healthy. And so in turn, that can also, it's kind of like a self-perpetuating feedback loop of doom. The next one here is deregulated nutrient sensing. And this one is centered around this protein called mTOR, or which forms two complexes, mTORC1 and mTORC2. So you have mTOR, and if you put mTOR with other proteins, in one case Raptor and others, and Rictor and others, you get mTORC1 and mTORC2. So these ones are at the center of a network of lots of different proteins that sense if there are nutrients around. There are others that play as well, like AMPK and sirtuins, but mTOR is kind of like the master regulator here. So what happens with mTOR? So well, mTOR, if there is lots of glucose or even essential amino acids, mTOR will in a way think that, oh, we are in a healthy environment, it's time to divide and maybe not focus too much on self-maintenance and repair. That's what it tends to do. Maybe that makes sense if you're like a single cell, like maybe if there's no nutrients, you want to wait and not replicate and just stick around and stay healthy. And when there are more nutrients, you want to spend more on replicating. In any case, mTOR, so if you upregulate mTOR and as we age with more blood glucose and that upregulates mTOR, if we do that, then that reduces lifespan, and that, as we will see later with a drug called rapamycin, that basically increases or the overactivation of mTOR increases the odds of basically everything that's bad about aging. It's kind of like a master of disaster in mTOR. There's also another thing here. So it's not just nutrients. There's also IGF-1. So we have this hormone called growth hormone that when it goes to the liver, it makes the liver generate IGF-1. IGF-1 then binds to the IGF-1 receptor, and that also goes into these pathways. So then again, you can find that in elderly people, well, actually in very long-lived people, they have like lower levels of IGF-1. So some people think that, well, maybe if your IGF-1 is dysregulated and you're expressing too much IGF-1, then that's also bad for you. However, there are recent findings that point to IGF-1 actually being good in our context. So, for example, when we go to sleep, we get a secretion of growth hormone. So, growth hormone, it goes in a periodic way. So we get a peak of growth hormone and then it goes down. And as we age, we get less growth hormone. So it might be that even though if you have like super high constant IGF-1, that's bad for you, maybe it's still good for you to have regular peaks of growth hormone and IGF-1. And as I said, this network of sensors that sense nutrients is implicated in, or it's expressed, or you can find it in every species, or in the ones that are usually used as models in the lab, like yeast, worms, flies, mice, and of course in humans. And they're the reason why the two most probably corroborated and robust interventions, which are caloric restriction and rapamycin. This is the reason they work. They target mTOR. And I will of course discuss these interventions in a later slide. So then we have mitochondrial dysfunction. So mitochondria again, so cells have different ways of making energy, but the most efficient way is through the mitochondria. And the mitochondria work by creating a protein gradient across their membrane. And so in a way, you can like build like a water reservoir, so to speak, and then that gradient of protons flows through a protein complex called ATP synthase, which then generates ATP, which is the famous energy currency in the cell, which the cell needs to do well everything, or many things. And so the mitochondria may get damaged for various reasons. So the mitochondria itself, all the energetic reactions taking place in the mitochondria can damage the mitochondria. Specifically, can damage the genome of the mitochondria. We also need to explain that the proteins that make up the mitochondria, some of them the mitochondria can make itself, or actually, so the genes for that are in the mitochondria. But for a few of them, they are in the cellular nucleus. So if those are damaged, if the cellular nucleus is damaged, then those proteins cannot be made, and they cannot get to the mitochondria. So the mitochondria is left without those proteins, and it gets dysfunctional. So in that case, mitochondrial dysfunction as a hallmark is caused by damage to mitochondrial DNA and loss of proteostasis. Mitochondria also play a role in apoptosis. So there's one hypothesis that leads to cellular death when there is damage, goes to the mitochondria. So if the mitochondria are damaged and a cell should undergo apoptosis, maybe it will not. It will stick around in a damaged state, which again is not great. Finally, there's another possibility that we have here, which is that we may also suffer from the expansion of defective mitochondria. The idea here is that that suppose that there is a mitochondria, and that mitochondria suffers a mutation or damage in its genome that shuts down these energetic reactions, that so-called oxidative phosphorylation, that is the process that generates all this energy. So in a way, it's like shutting down the nuclear reactor. So there's no more damage. And then suppose that some other mitochondria in the same cell, they kind of die and stuff. So when mitochondria are damaged, the cell will phagocyte them. They will just undergo autophagy. They will get eaten, broken down, and recycled. And the other mitochondria will then expand to cover for the mitochondria that died, to replace that. However, if if the other mitochondria, if they're getting damaged and replaced, but the mitochondria that are not working, they're not getting damaged, like they're never getting damaged because they're not doing their job, they're not generating energy. It could be that those mitochondria, by natural selection, end up proliferating more than the others, and then you end up with cells full of mitochondria that are not doing anything, which again doesn't sound great. Now, this is the extent to which this matters and how this happens are still a matter of speculation. But I just like to mention all the various ways in which mitochondria can be nasty for you if they are not working properly. The next one is one that I've mentioned earlier a couple of times, which is cellular senescence. So cellular senescence was discovered in 1961 by Hayflick, and this is the idea that well, when the telomeres shorten initially, although other things can cause senescence, when the telomeres shorten, or you get overactivation of some genes that may lead to cancer, or when you have damage, for example, from radiation damage, the cells go to a state called senescence. Now, in the senescent state, the cell is still functional, still doing its thing, but it's also secreting this thing called SASP, or the senescence-associated secretory phenotype, or phenotype in the slides. And this leads to inflammation and cancer if there is too much of this. Now, the idea of the SASP, there is still debated exactly what the issue with the SASP, but if you have like one or two senescent cells, your immune system will come and clear them out. So this inflammation coming from the SASP is not necessarily a bad thing. Inflammation attracts the immune system to clear up debris and other things. So that's fine. However, if there are too many senescent cells and the immune system is weakened, then that SASP can also cause damage that can lead to inflammation and cancer. And so, for that reason, because senescent cells are they are meant to, it's okay if they form, but they are meant to be removed. If they cannot be removed, we can try to remove them manually. And so this can be done with a class of drugs called senolytics. So senolytics, they are currently undergoing trials in humans. So there's this company called Unity that is working on that, among others. And this goes, it has been a few years to get to this point. So it was like, in 2004, it was first shown that senescence could reduce in some way with caloric restriction. Then 4 years later, we learn about what this SASP was exactly doing. 3 years later, we say like, hey, what if we kill these cells? If the SASP is a bad thing, if we kill these cells, do we get an improvement in mortality and other things? And says yes, in mice. That was in 2011. And then finally, 8 years after that, we go to the point where we already have these drugs in clinical trials. So then we have stem cell exhaustion. So senescent cells are amongst a few cells that divide in our body. So most of the cells in our bodies are so-called post-mitotic. They don't replicate. And I say most because it's kind of tricky. So if you take, for example, fibroblasts from the skin, fibroblasts don't divide normally. If you have a wound, they might divide. If you put them in a petri dish with no other cells around, they will also divide. And this is so-called because some cells are also subject to so-called contact inhibition. So even in a mechanical cell, if cells have too many cells around them, that will also inhibit their growth. But in general, we rely on these stem cells to replenish cells that get damaged and die. So senescent cells, the idea is that they are there and then they divide into another stem cell, and one cell that will eventually differentiate a bunch of times and end up becoming like a neuron or a liver cell or a skin cell. Or blood, white blood cells, whatever. There are many kinds of these stem cells. And well, so the issue here with the stem cells is that, well, the cells themselves can get damaged. But also the place where the cells are, so they are not less in random places. They're in this place called the microenvironment, which is a special place in various parts of the different organs. That is meant to keep the cells kind of happy and functional. If that degrades, you can also see a loss of function in the stem cells. And in the last decade, we have seen a shift from the idea that senescent cells themselves age and get broken and get damaged to the idea that maybe the cells are okay, and it's the environment that makes them be dysfunctional. So in that case, if you improve the environment, maybe you get back the function from the stem cells, and you see, for example, experiments where you take stem cells from like all individuals, you put them in young individuals, and then the stem cells seem to be fine. Um, so that's it for the stem cells. Yes, another thing to mention though. So stem cell therapies, there are a bunch of clinics that offer stem cell therapies, but the stem cell therapies are kind of difficult to do. One is that if you do it wrong, so stem cells have cells with lots of potential for division, they can lead to cancer potentially if you don't do it right. And also because probably the best way to get stem cells is from your own body. As in, if it's from someone else, you may get some rejection. And the best way to get them from your body in a cheap way, or in a safe way rather, is in a non-invasive way, is to take, for example, some cells from your skin, and then with some clever reprogramming, you can make them into induced pluripotent stem cells. And with those cells, you may be able to then use them for therapy. But this is not that advanced yet. So stem cells, again, it's like telomeres. It sounds like maybe a few years ago, like stem cells are going to be this huge thing, and they may well be. They are indeed a key part of the healing process. But it seems that progress is not going in this one area as fast as people initially thought, that there are more things that one has to address to fix aging. The next one is altered intercellular communication. At first, it may seem that aging is just aging of a cell, and then just like aging of the body is like aging of a bunch of cells, but that's not true because, well, cells talk to each other. Cells are in specific positions in the body. So, for example, if you have like inflammation near your gum, in your mouth, that may affect your brain because it's just very close to it. So we see altered intercellular communication through a deregulation in insulin response, as we saw earlier in the nutrient sensing case. Also in the increase of inflammation. So inflammation, again, is this process that in an acute temporal way is helpful for the immune system to clear up damage. But chronically, you get this high level of cytokines, and it kind of like tells your body that you are constantly being invaded by something. You get this state of inflammation, which is telling your immune system to be constantly activated, and that can lead to damage. And in turn, well, inflammation also in turn leads to disease. Another thing that we can say here is that the extracellular matrix, which is the thing that's between cells. So in between cells, you have a mixture of elastin, collagen, hyaluronic acid, and other things. And as we age, the thing stiffens; it becomes more rigid. And this may make it more difficult for T cells to get to dendritic cells. So the dendritic cells are the cells that tell the T cells, hey, we found a virus, or we found this thing, you should go and eat. And if the T cell doesn't get to the dendritic cell, then it doesn't get activated. And then you get an impaired immune response, which is not great. Finally, we have the problem of blood-borne systemic factors. So this is the idea that proteins and hormones that are present in the blood regulate the function of the body. And as we age, proteins and things that should not be in the blood are such amounts, they just keep accumulating as we age. And conversely, things that should be in greater amounts, so when we are young, decrease in amount, leading to disease. It's kind of like telling the cells, hey, we are old now, you should be old. It's kind of like that. We then have immunosenescence, which is the aging of the immune system. And if you're interested in this, I recently wrote a whole review on immunosenescence that you can go check from my blog that was in the first slide. So the idea here is that the immune system becomes less able to respond to infections, vaccines, or cancer. The immune system is composed of loads of cells. You have B cells, natural killer cells, T cells of various kinds. You also have these dendritic cells, macrophages, and so on and so forth. And figuring out exactly what goes wrong with the immune system is very difficult. So there are many things that change with age. So for example, here's one thing. So as we age, we have less TCR diversity. So TCR is the T cell receptor. So this is the thing in the T cell that helps them recognize specific antigens or patterns from viruses that may be a threat. And if you have a lower diversity, it's less likely that you're going to have a T cell for that specific virus. Likewise for naive T cells. So naive T cells are T cells that have the capability of they have not been activated yet by the dendritic cells and can expand a lot to address these new threats. And there's also a decrease in just like the overall level of lymphocytes, it just goes down. Just like they can be naive, they can be non-naive, whatever they are, just the whole thing goes down. And interestingly, at least if you take the ratio between lymphocytes and another cell from the immune system, another cell type called monocytes, if you take this ratio, that is predictive of cancer, atherosclerosis, and all-cause mortality in general. So there is probably something going on with that. Now, to what extent? There has been argued that maybe these things don't matter that much. Maybe it's not that we have less diversity of TCRs, or maybe that, or even maybe the cells themselves, maybe they are fine. So some people argue that maybe the cells are less effective. But it can be that it can be many things. So it can be that the T cells are fine, but if the dendritic cells, or the DCs here, if those cells are impaired, or if the T cells can't get to them, then even though the cells are fine, then you don't get that activation of the adaptive immune system. Hence, as with the case of stem cells, we have seen also a shift from the idea that the cells are broken to the idea that things that are extrinsic to the cells may be addition. In this case, the environment in which the cells operate, that may be to blame in this case. Something else which seems to have changed the views in the field over time is the role of cytomegalovirus, or CMV. So this is a virus that's a herpes virus, which means that once you get infected, and by the way, around 80% or more of the whole population of most countries are infected. It's kind of everyone is infected with this thing. But it doesn't really have symptoms if you have a competent immune system. So if you're taking immunosuppressant drugs, then this CMV may become active, and then it may be a problem. But mostly it's not an issue. Now, some people thought that initially that because you have this virus constantly being kind of active but not too much, your immune system was going to constantly be trying to fight this infection and kind of getting exhausted and trying to generating cells that are specific for the CMV, so-called memory inflation. So constantly dedicating more and more of the pool of T cells to fight CMV until there are no more or reduced amount of T cells to fight something else. Thus, this would place CMV as a key cause of immunosenescence. But now it seems that the idea is that this we don't get this huge increase in cells specific that are targeting CMV. We instead get something like a one-off increase and then stay stable. So if people get sick and they have CMV, this may be because the immune system was also weak. It wasn't probably the CMV. So again, in this case, it may be that altered cellular communication might be the key to why we're undergoing immunosenescence. One other thing that I mentioned earlier that may also play a role here is the involution of the thymus. So the thymus is this gland we are born with, and the T cells are created in the bone marrow from hematopoietic stem cells. And then they travel to the thymus, where they are trained to both recognize pathogens and also to not to recognize our own cells. So because if not, it will attack ourselves. We suffer from autoimmune diseases. So if the thymus shrinks, then these T cells are not going to be properly trained. So the odds that they're going to be attacking your own body may increase. And likewise, the odds that these cells are not going to recognize properly pathogens are also going to increase. The evidence to whether or not the thymus matters is, again, it's kind of contended or debated. There is some recent work we'll talk about later that addresses this question in humans, which sounds very promising, but it does seem like the role that the thymus play may also be not so much in producing new naive T cells, but also in some sense in also orchestrating the immune system in more of a regulatory way rather than just like raw amount of these cells. We have inflammation. So inflammation is that topic that pops up everywhere in aging. So it's like, oh, this leads to inflammation, and inflammation leads to all these things. So you can see at the bottom right this picture that reflects this idea that everything is kind of connected to inflammation. So the idea with inflammation is that where this inflammation is coming from first is that as we age, we are, maybe because of the immune system is weakened, we are exposed to more viruses. Our skin also degrades, so the skin is the sort of biggest organ, and that's a barrier against pathogens. So if your skin gets literally thinner, you may get more of those pathogens into your body, which will then trigger that inflammation to react to those pathogens. Likewise, in your intestine, it may be that the junctions between enterocytes, which are the cells that are lining the intestine, they if they kind of either die or if these junctions are not perfect, if they open up, the bacteria from your intestine are going to leak into your enterocytes of the body, and they are bacteria. After all, so the immune system will also trigger an inflammatory response to tackle them. And then finally, various other things sadly also induce inflammation. So first we have receptors called TLRs and or toll-like receptors or NLRs, which are meant to detect common patterns in bacteria. So these are part of the innate immune system. So they're like generic things to detect pathogens. But they also react to things like saturated fatty acids or components of dead cells. And also mitochondrial DNA, because mitochondria are bacteria. Suspiciously, well, they came from bacteria, so their DNA, it may look like bacterial DNA to the immune system, so it triggers this inflammatory response. So we get this release of various cytokines. These are these proteins that trigger the immune system to actually get activated, and that in turn even that leads to basically everything that's bad it leads to. And we can see this from RCTs. So from RCTs, we can see that, okay, if inflammation is mediated by all these cytokines, like IL-6, IL-1, beta. If we block those with a monoclonal antibody, we can actually take those out from us. If we do that, then we get lower odds of cardiovascular disease, Alzheimer's, cancer, diabetes, and all-cause mortality. Meaning that actually there's something going on with that inflammation, that our body is overreacting to all that damage. And finally, I think it also seems that the recent papers seem to point to the role of the environment a lot, in the sense that they claim that, well, some people don't get all of this inflammation and other people do. So it may not be a universal hallmark, and it may only happen in conjunction with other things that you may be exposed to. Next topic in line is NAD depletion and sirtuins. So sirtuins are, they are proteins that they use NAD, so they work with NAD to regulate the epigenome, among other things. So they regulate the epigenome in the sense that they work to silence genes. And they also have a role in repairing DNA. Now, this sounds like great, like you want those sirtuins to be active, and indeed in experiments, if you have more active sirtuins, you do get more lifespan. And some people have proposed there's some compounds, so-called sirtuin activators or stacks, such as resveratrol, petrostilbene, or SRT2104, which is from this company called Sirtris. And this all purports to activate sirtuins. The extent to what they do, again, it's debated. Resveratrol initially didn't seem to work in mice. There is this debate that the dose wasn't high enough, or that you need to take fat with resveratrol to actually absorb it properly. In any case, sirtuins are for sure having a salubrious effect on longevity. Now, as we age, NAD goes down, and sirtuins need this NAD. Now, why does NAD go down? Well, initially they thought that, well, maybe it's DNA damage or something that is causing the sirtuins and other proteins called PARPs that also repair DNA to be overactivated, and that's like consuming all the NAD. But actually, no. Actually, it's inflammation. It seems it seems to be that senescent cells, which generate inflammation, or inflammation from other sources, is causing macrophages, which macrophages can be M1 or M2, and you can read my immunosenescence article for more information. These macrophages that express this enzyme called CD38. This enzyme takes NAD and it's very efficient at taking NAD and making into NAM, which may be to try to reduce inflammation, but this thing is so effective that it just depletes NAD in your entire body. So it may be that in this case the issue with NAD depletion was inflammation all along through macrophages. So maybe if we deal with inflammation, then we also solve the issue of NAD depletion. Now, of course, if you have NAD depletion, you can also try to supplement NAD. So that, there are like NAD supplements like nicotinamide or nicotinamide riboside or NMN, which can be supplemented to increase NAD levels. This doesn't seem to increase lifespan, or it does in an inconsistent way. It might do maybe it does in flies but in mice it's less clear. In mice, as you can see this experiment here, in mice, you can see that the author of this paper found that you increase health span, so you make the mice healthier. So, for example, in this case, you reduce blood glucose area under the curve. So this is a test where you give glucose, literally you stop glucose with water to mice, they drink it, and then you measure their blood glucose, and you see how they react. So ideally, they will react quickly, and insulin will react, take the glucose into the body. It won't let the glucose float around in the blood. And so reduced area under the curve means that insulin is being more active. So what this means is that relative to control, which is the blue one, the animals that were fed NMN or a higher dose of NMN had a lower area under the curve, meaning that they had an improved glucose metabolism, which in and of itself, it's already a good thing. So even if they didn't live longer because perhaps this thing is not addressing something else that it's still killing the mice, you're still getting health benefits. And again, because NAD is a central part of cellular metabolism, it's helping across the board. It's not just targeting, in this case, with diabetes. It will help everything effectively, everything that is using NAD. The brain also plays a role. So it might seem at first like the brain just kind of sits there and does funny things like making longevity slides, but it also does other things like regulating hormones and the circadian rhythm, and even adipose tissue. Like the brain can make you fat to put in a funny way. So in this case, we know that the brain is probably up to something, because when you rejuvenate the hypothalamus via stem cells, you get improvements in various things. So here I want to draw your attention, well, first to the picture at the bottom. So in this case, I-kappa B alpha, in this case, they are neural stem cells. So the red line represents stem cells that were selected for their ability to resist inflammation, while the green ones are stem cells that do not survive inflammation. So there's inflammation in the brain, and if we put regular stem cells in there, they are not going to be effective. So we will put stem cells that can survive inflammation. We'll put those in there. So this gave them, and they did this in mice that were already kind of old. They were already maybe 700 days old. So you can see that their lifespan increased by a non-trivial amount, both median and maximum, which is something. But not only that. So if you look at the chart at the top, you can see that they also tried other things. They tried injecting things just like vehicle, just like saline, or astrocytes, which are a kind of cell that you can find in the brain, or other kind of stem cells, or these special stem cells that would resist inflammation. And only these ones you were able to see improvements in like how much they can run, or some measures of muscle endurance, or locomotion, how much they can keep walking. And some measures that maybe are more subjective, like sociality or knowledge recognition index, which again, you could argue that you can do the tests you want to get the nice results. But I think the test that can your rat run faster and for longer, that I think it's pretty significant and objective. And so this means that those stem cells in the brain are doing something in there. Conversely, you see the opposite. So if you remove the stem cells from the brain with a virus that targets them, you get a reduction in lifespan. And how does this work? I mean, what is the brain exactly doing? Well, it might be that it's through our hormone regulation. It might be that it's growth hormone that is doing the job here. It may also be that it's through regulation of the of other stem cells. So in our bone marrow, there are nerves, and so it may be that the brain is actually affecting the way in which those cells are expressed. So the brain is not just like it's kind of like the world's manager. It's like I'm trying to micromanage everything. So it's trying to regulate everything in our body. So it may be that faulty signaling from the brain is causing stem cells in your bones to not be effective, which is kind of like wild. It's kind of like something just like up down there in your bones is being impaired because of your brain. Which again, so maybe if you're going to rejuvenate those cells down there, you need to fix the brain first. Which again, this is very hypothetical, and the fact that the brain plays a role seems more or less clear. I think that how and when and where it plays a role, that is less clear. So next thing in line we have here is the aging of the extracellular matrix. So this is the thing that I mentioned before that's in between the cells. So we have everything from elastin, collagen, hyaluronic acid, but also just like fibrosis. So you can guess this like just fibers, but not only that. So in these things that are in between cells, they can get cross-linked. So they can get like stuck to each other, or you can get proteins in there that just like get them sticking to each other. So some of which are called advanced glycation end products or AGEs. This makes this unit more stiff, and stiffness for purely mechanical reasons, it leads to senescence in cells. So it's quite interesting. So if you take a cell and you put it in a stiff extracellular matrix, it will become senescent. If you put those cells in a younger ECM that's like more flexible, then the senescence will reverse. It was previously thought that senescence was irreversible, but the ECM apparently is regulating senescence to some extent. Not fully though. I mean, the cells you put in the young ECM, they eventually undergo senescence, but it seems that a stiff ECM is prematurely making them senescent. So, and if we make this less stiff, besides getting less wrinkly skin, we may also get other things like less inflammation. It's also related to cardiovascular disease. So these things are also forming in your arteries. So if your arteries become more stiff, then it will be more likely that either they will break and leading to blood clots or strokes and other things, so which are not great. And also, I just have to say that yes, I also mentioned earlier that this can influence the immune system because of the reason I mentioned before. This is not as well studied as the other things. So many of these things are not even like proteins. These are just like, or rather, these are not like biologically active. They're like sitting there. It's like collagen is meant to be more like a scaffold. It's not meant to be interacting a lot with the rest of the things in the body. And this particular thing seems particularly difficult to improve. The reason being that the most common age is glucosapine, which is, well, we cannot break it down, it seems. Then second, it's difficult molecularly because of its structure. It's difficult to break in the first place. There are, however, some interesting things here. So first, we have been able to synthesize glucosapine in labs a few years ago. You may be surprised how late this was, but that's it. And there's also a startup called Rebel Pharmaceuticals that they are trying to develop precisely glucosapine breakers. Another thing that I also want to highlight here is that if you want to do these studies in mice, glucosapine is not as relevant. So glucosapine is the most common age in humans, but not in mice. So maybe glucosapine makes sense if you live longer, you accumulate this thing. So it may be that it's going to be more difficult to do studies in mice because they are not like us with regard to glucosapine. Next thing is reactive oxygen species. So these are molecules that have oxygen in them that are highly reactive. So they would react with lipids and proteins and other things and wreak havoc all across the board. Now, back a few years ago, there was this theory called the oxidative stress or free radical theory of aging. There are like a bunch of theories that have this name. And likewise, there is a mitochondrial free radical theory of aging. There are a bunch of these. So it was thought that, well, maybe so these things, also to clear, these things are in high concentrations very damaging. In fact, the immune system cells like macrophages will use high concentrations of ROS to kill bacteria. So they are obviously harmful in high concentrations. And so the thinking was maybe mitochondria are making too much of this, and that's leading to disease. Well, it was later found that the evidence was quite inconsistent. So maybe mitochondria are not even a net source of ROS. And also knocking out antioxidant enzymes do not seem to improve lifespan. And also giving antioxidants to mice also doesn't seem to do much for them. Well, it was later found out that ROS plays a role in signaling. So cells use ROS as a signal. So when you exercise, you get more of this ROS. And so, for example, if you exercise and you take an antioxidant like vitamins, that's going to reduce the good things that exercise is doing for you. It's kind of like hormesis, when a small amount of a bad thing is actually good for you. Now, the extent to which ROS matters is unclear. Some people say that this theory that has been convincingly refuted and it's not worth thinking much about them. Other people still swear by the theory that it's totally valid. I would personally think that they matter to some extent, but they are not the core thing. Again, just like telomeres and stem cells and other things, this comes from the age where people had like this is the one thing that's causing aging, and now we are moving towards a model where it's many of these things and it's extremely context dependent. Because, for example, you can delete all of these. Some people argue that sure, just because you delete all these antioxidant enzymes doesn't mean that ROS doesn't matter, because you still have the key antioxidant enzyme that's called superoxide dismutase or SOT1. That one is still active. So if you delete that one is harmful. So it might be that one gets overactivated, and in that case, you don't get to those dangerous levels of ROS. Also, some people argue that in species that are long-lived, you see decreased levels of ROS and so on. So again, this remains unclear to what extent we have this. In any case, it seems that if we fix mitochondria and if we fix DNA damage, then we may get less ROS, and so this will also go away as a problem. And we have another one of my favorite things here, which are epigenetic clocks. And you can find also on my blog, I did a review on this specific topic. So epigenetic clocks, the idea is that we can take these CPGs from the epigenome, and then we can train a very simple model. In most cases, it's an elastic net, which is a fancy linear regression with some priors or penalties. And then based on these methylation patterns, we can say, okay, we want to train this to predict the age of the subject, or we can predict if we have like cohort data for if we have like later measurements, we can predict how long they have until they die. And these clocks are surprisingly accurate. They are, I think, more like 80 or 90% accurate in some cases. So if I measure your epigenome, I can tell you your age, maybe plus minus, I don't know, if 1, 2, 3 years or something like that. These clocks were initially kind of controversial. So at first, they were developed to predict age, and then they were tested on interventions like rapamycin. So rapamycin I will talk about in a moment, but it's a drug that has been shown to increase lifespan. And so in theory, if you have this clock that's measuring age and it doesn't go back when you rejuvenate an organism, that's kind of weird. Right? So then they develop second generation clocks which measure time left to lifespan. And these ones do work. These ones show, and you can see it on the right. These ones you can see the difference between the actual age and the epigenetic age in different cases. So for example, you can see at the top right that in this case, wild type mice of the UMHET3 strain If you compare the wild type with the rapamycin ones, they are I think it's usually like 10 days or months. I think it was months younger than they would otherwise be, and likewise with caloric restriction. Similarly, there are these other mice that are called dwarf mice, which are smaller. They have a lower expression of IGF-1, and they live longer. And in these ones, you can also see that these mice have a reduced epigenetic age acceleration, so they are younger than they should be for their actual chronological age. And you can also, in this case, these ones are predictive. So if you have a high score, so if the model is saying that you are old, or you have a high score, in this case they call the score afraid. If you have a high afraid score, in this case it's mice, but you're going to die sooner than otherwise. And the thing is that this, because you can see that mice that are 20 or 26 months old, they are already kind of old. But if you read these papers, you can see that you can replicate this thing across the whole mice lifespan. So you can do the measurement when they're young and when they're adults and old, and you still keep seeing that these epigenetic clocks keep telling you who's going to die earlier or sooner. Now, the good thing about these clocks is that we can use them to accelerate research. So if I have this intervention that supposedly is going to decrease mortality, you can just use the clock and say, okay, it actually worked. It's reversed aging. Or no, you don't have to. May not have to wait for like 10 years as if it worked and wait until people actually die. But there's still some work needed to validate and incorporate them into actually discovering drugs, because there are like lots of clocks being made. Different people. Some people are using neural networks or random forest to train these clocks. So there's still some time to go before we can say use them like for the FDA, so they can approve drugs using this. So if you're more interested in the clocks, as I said, you can go to my website and you can read my review in length about this stuff. Okay, so we finished with the things that or the different hallmarks of aging, all the things that happen as you get old. Now we get into what can we do to actually slow down or revert aging. Okay, the first one is rapamycin, which is probably the best known intervention there is, and rapamycin, as I said, downregulates mTOR. Now mTOR is just, if you downregulate mTOR, you get two things. You get increased autophagy, so your body will recycle more often the different components of the cell, and second, it will reduce protein production. In a way, if you're producing fewer proteins, the odds of producing misfolded proteins are perhaps reduced. So probably the combination of those two things are getting you improvements across the board. You're getting the cells are doing what they're supposed to do, and this is all the cells because all the cells have mTOR. Now, so rapamycin, it's typically used as an immunosuppressant. So if you take a high dose of rapamycin, a very high dose, your immune system will become ineffective. So this is used, for example, to avoid rejection of an organ transplant. However, so this is why initially people were like, oh, rapamycin is so great, but it causes the immune system to kind of be suppressed, so we need to develop an alternative. So some people were looking for rapalogs, which are rapamycin analogs that kind of behave like rapamycin, but without having this immunosuppressant activity. The thing here, though, is that later we find out that actually it's not that bad. If you have like lower doses of rapamycin or pulse, as in taking rapamycin periodically, not every day, you actually get the good effect. So it turns out that rapamycin starts by inhibiting mTOR1, which is the one that's responsible for all this stuff. And if you keep increasing the dose, it goes to also inhibit mTOR2, and you want to stay away from mTOR2 to avoid these other effects. So rapamycin is a drug that you need a prescription to get. There are some doctors that will prescribe it for longevity, but definitely it's something that extends lifespan maximum. I mean, lifespan in anything you can take in life form, you give it rapamycin and it lives longer. So it's pretty much it's a very solid intervention. I guess in humans, the common side effect of rapamycin taken again at a low dose is ulcers in the mouth. But again, I'm not expert in rapamycin in particular, and I'm not recommending anyone to start taking rapamycin, although some people say do recommend, yeah, that you should like I said, that the moment like it's never too late to take it. I won't enter in a debate. I'm just saying that it works and how it works, and that's it. Well, next thing we have caloric restriction, which is just like eating less food, or in some contexts, eating intermittently. The important thing is introducing some amount of either fasting, some measure of nutrient deprivation, so we can actually go down and down-regulate mTOR. So you can see in these pictures how in pictures A and B, you can see monkeys, and this again, this is even closer to us than mice. You can see monkeys that have been calorically restricted, and in C and D are the CR ones, and A and B are the regular ones. And you can see that the C and D ones look healthier. Not only they look healthier, but also their average lifespan was increased by the number of years. There's still some debate. So there were two of these studies, and there is still it was debated about to what extent this increased maximum lifespan, because the studies were not exactly the same, and the diet was not great, and other things. But I guess the consensus about these two studies is that it shows that in primates that are kind of like us, and this study lasted, as you can see, for decades. In primates, caloric restriction works to increase median maximum lifespan and health span. And also, if you look at humans, if you look at, for example, trials like the calorie trial in humans, or Okinawa ones in Japan that eat a smaller number of calories, or you look at the Caloric Restriction Society, which is a society of their members undergo caloric restriction, you see improvement in biomarkers, which I think this evidence, together with the evidence in primates, I think strongly suggests that this works in humans to decrease mortality and increase lifespan and health span. There are other interventions here that work. I mentioned intermittent fasting to some extent, and also, and for the same reason, if you decrease intake of methionine, which is an essential amino acid. In fact, methionine codes for the it's amino acid that starts every protein, which is it's a pretty much essential amino acid. If you restrict that, which is found usually in animal products, which may explain why some benefits of more vegan diets. If you restrict methionine, animals also live longer. Next one I talked about in the context of senescent cells. These are senolytics, which have been tried in mice in this case, and you can see that besides improving function, they also increase lifespan by, in this case, around 50 days of mean lifespan, and maybe less than that for maximum lifespan, because again, something else is going to be killing those mice, not just the senescent cells. And you can see that promisingly, these mice were treated when they were already quite old, and still they got some benefits there. And not only got some benefits, because this study also looked at not just how long they lived, but also physical function and other metrics. So it's not just how long you live, but also how healthy that lifespan is. This has also been tried in humans recently. So this was in trial. This is from there's not even a paper for this. This is from a slide deck from Unity, where they show that compared to placebo and in a dose-dependent way, their senolytics can decrease a composite metric of arthritis. So they can decrease pain and increase function in quite a significant way for arthritis. And as I said earlier, this doesn't mean that this is ineffective or not, or it's not going sufficiently. It's not going to removing all senescent cells. This means that there's probably something else causing arthritis, and this is not targeting that. But again, it's very promising. It's going through clinical trials, and interestingly, this same approach, Unity is also working on this same approach to be applied for cataracts, for eye disease, and other conditions. So it looks very promising. We then get to one of my favorite papers. This is a paper from Alejandro Campbell from the Isabel Monte lab from the Salk Institute. And what they did here is they took, in this case, these were not wild-type mice. These were mice that have accelerated aging. And they genetically engineered from those mice, they genetically engineered some that when they are fed doxycycline, which is an antibiotic, when you do that, they express a series of reprogramming factors. So these are the same factors that you can use to make cells into induce stem cells. And if you just put these factors into mice, they just develop tumors and die. But if you express them in a transient way, if you don't go all the way, if you do it only a little bit, these factors kind of have the ability to actually rejuvenate the epigenome. They can actually replace back all the and remove the methylation that was kind of damaged. It can revert that damage. So cells have a way of actually knowing what methylation should be in there and can undo the damage. So on the graph on the left, you can see that the lucky mice, is that the regular mice without any genetic engineering, regardless of whether or not you give them the doxycycline, they live more or less the same. The genetically engineered mice, lucky for F, they live a bit longer. So maybe it means that they are kind of expressing these genes a bit. But when you give them the periodic cyclic doxycycline treatment, their lifespan is greatly increased with respect to the wild type. It's increased by, if we compare it to the wild type, almost by 2x, and same thing goes for maximum lifespan. So this might be one of the most effective interventions ever there is. And if you look at the paper, look at many things that improve. So you can look at, for example, the second picture looks at glucose metabolism, which again, glucose matters for diabetes and for this the formation of this advanced glycation end products. Well, you can see that with the doxycycline mice have a better insulin response with a lower area under the curve. And likewise for senescent cells, you can see that wild type have like 20% of senescent cells. And of course, these mice that have accelerated aging, they will have more senescent cells. So they started with like 60% of their cells in a sample that they looked at being senescent. 4 days after the treatment, that had gone down to like 30%. Again, this looks extremely promising, and there's already a startup called Turn.bio that's looking into this, because the epigenome is upstream of all these sort of things. So so apparently with fixing this, you can potentially fix senescent cells and loss of proteostasis and glucose metabolism. You can fix all of those things if done well. Because again, if this is not done properly, you're going to get very nasty cancers. So that's it. This has not been tried in humans, so we'll see when we get there, but it looks extremely promising, I'd say. So likewise, this can be used in different ways. So in this paper from the Sinclair lab, they used it to improve ocular function in mice. So what they did was two things. They induced an injury in the nerve of mice. They crushed the nerve, and they also introduce some microbeads in the eye, which increase pressure in the eye, which leads to damage. First, they're looking at axon density of the optic nerve, which is a measure of, of course, if you have no axon density in there, you're not going to see anything. So it's a measure of how healthy that optic nerve is. So usually you have like, if you just inject saline, which is like inert, whether with salt, you have like 50. When you inject these beads and you induce this damage, you get down to 40. Now, if you put these beads in there and you add PBS, which is and it's meant to be also like a control, it doesn't seem to do much. And in the same mice, when they without expressing or prior to expressing these reprogramming factors, so-called in this case, the genes are OCT4 and SOX and KRAS, OSK. When they actually get to express these genes, the nerve actually physically regrows. And they actually look at the nerve itself. It actually regrows and connects itself. And not only do you see the nerve growing back, but you also see an improvement in ocular function, which you can see in the graph on the right with the test that they did, which again looks extremely promising. And also in this case, they did no genetic engineering. What they did was to use an associated adenovirus. Actually, they inject the virus, and the virus infects the cells and makes the cell express these factors. So again, it shows another possible use, in this case to restore vision, that epigenetic reprogramming may have. I hinted earlier at the thymus. So the thymus, again, this gland we have here, it plays a big role in the immune system, and some people believe it's key to improve aging, to improve the aging process. So in this case, this was from a paper from Greg Fye and others, and what they did was to give patients a combined treatment comprised of growth hormone, DHEA, which is and metformin, both those two, and in part two, because growth hormone might induce increased blood sugar, and these two counteract the increased blood sugar, and zinc, which seems to also have an effect on the immune system. So these treatments show first, and you can see in the picture in the center, that TFFF means thymus fat-free fraction. So the thymus, it's as we age, it becomes fat, and this shows basically if you had like 60%, means that 60% of the thymus was not fat; it was a healthy thymus. 10% was more that the thymus was mostly fat. And what this shows is that for those people with a thymus already more or less healthy, maybe only half fat, didn't do much. For those people that have a very shrink thymus, it almost doubled the size of the fat-free fraction in there. This was accompanied by an increase in the lymphocyte to monocyte ratio. It came from 4 to around 8, and if you want to what does this mean? So, for example, I myself a more or less healthy person, and in my latest blood analytic, my lymphocyte to monocyte ratio was maybe around 8. So it's a huge improvement. It's basically making this ratio the same as it was when they were young. Now they didn't fix anything though. So they looked only at a few things, and it was a very small trial with maybe around 9 people. They for example looked at senescent T cells, and that didn't have an effect on that. But perhaps the most interesting thing, or the reason this paper got so famous in this field, is that they used the best epigenetic clock there is, which is group age. And they showed a persistent decrease by 2 years in this epigenetic age curve. So this might be interpreted as increasing lifespan by 2 years in 1 year of treatment, which they claimed was the first ever confirmed case in humans of age reversal. Now, this has not been confirmed yet, because again, clocks, the science behind epigenetic clocks continues to be debated. And it may be that this treatment is improving some parts of the epigenome and not everything. So maybe the parts that it is improving some, but not the others. So maybe this is not affecting, or this intervention may not end up affecting mortality at the end of the day. Nonetheless, it looks quite promising because it shows that one, you can actually reverse the involution of an organ without having to do like a whole transplant. Second, it's improving a metric like lymphocyte to monocyte ratio, which is associated at least to many diseases, and it's showing promise with the epigenetic clocks. So again, very promising. And also there's of the paper, also well, promising because it was only humans. This is not mice. This is actually human volunteers, and they're working on another trial called TRIM-X, where they are looking to increase the number of things they're going to try, and they're going to have more volunteers, and they're going to be looking at more things to refine this treatment. Another one here, actually, you probably have heard about heterochronic parabiosis, aka blood boys. This is the idea where you take, or this was initially found where you say, hey, what finds if you take an old mice and a young mice, and you seal them together so they share the same circulatory system? Well, the young mice seem to live longer and the old mice seem to live longer and get healthier, and the young mice seem to kind of get older or something. So it seems like there's something in the blood that gets sealed. So also some people thought, well, what is it in the blood that is doing that? And so some recent papers have looked at what that might be. In this case, there was this one paper. What they did was not a blood transfusion. This was done in mice, and what they did was to derive a proprietary plasma fraction they call elixir. So we don't know what it is, and they injected this into mice a bunch of times. So there was no blood exchange here going on. They just injected. They didn't take anything out. Just injecting this plasma fraction, which is meant to mimic the plasma that's present in young mice. And for record, plasma is if you take blood and you take out red blood cells and white blood cells, you're left with plasma, which is saline with a bunch of proteins, hormones, and some nutrients. Okay, so you give the mice this plasma, and you get a decrease in IL-6. IL-6 is a marker for inflammation. You get a decrease down to the levels of younger mice in just a few days, and this thing persists after a single treatment for half a year. These are days. Likewise, you see a decrease in circulating glucose down to the level of young mice, well, rats in this case, And for cholesterol, total cholesterol is the same thing. You're also seeing a decrease in cholesterol by a huge amount down to the levels of young mice. They also looked at epigenetic clocks in this case, but they use clocks that are not as validated as the GrimH, so I haven't put those pictures here. And I think that these three things are probably better indicators that this trial is doing something good than looking at the clocks in that case. But if we want to believe the clocks, they seem to have reduced epigenetic age maybe by 50% or so, but also a huge margin. They didn't do a lifespan study in this one, but they are working on that. So we may see soon, we'll see if this proprietary replacement fraction actually did something. Another thing they have looked at is to say, hey, what if it's not something in young mice? What if it's something that accumulates in old mice? That's bad. So in this case, they are taking out plasma from mice and replacing it just with saline. It's just like salty water and albumin. Albumin is like, we can think of like nutrients, nothing fancy. And actually, this therapy is already approved by the FDA in the US for humans for some conditions. So it's nothing strange, right? It doesn't sound like it should do anything weird. So in these graphs, what you can see here is you can see different tissues: the hippocampus, liver, and muscle. And YY means young mice that just exchange plasma with young mice. OO, same with old mice. And the interesting ones are the ones in the center. So YNBE means taking young mice and then taking out their plasma and then putting this prepared fake plasma, which is like saline and albumin. And ONBE is old mice where you do this treatment. So again, no blood exchange, just taking plasma out and putting plasma in. Okay, so what do we see? Well, the first picture shows number of Ki67 positive cells. So this is a marker for cellular division, which in the brain it means how active the stem cells in the hippocampus are. And you can see that they're almost not active at all in the old mice. Now, in the old mice, when you just like take this thing out of their blood, that goes up almost to the same levels as the young mice. Likewise, looking at the liver, so the liver becomes fat as we age. And as you can see in the old mice, they are taking their fattiness as 100% as a baseline. In young mice, that's maybe like 10% or something like that. And if you do this blood exchange, you can see an example of maybe it goes up to, in one lucky case, up to 50%. So you're like decreasing it by half the deposition of the liver. And not only that, if you look at the next one, at fibrosis is an increase in connective tissue in between cells. So instead of having liver, you just have scar tissue instead of having liver. So I guess you can see that these old mice, when in the ONBE case, it goes down very close to the young mice. Likewise, when you actually induce an injury to the mice, when they actually harm the mice in the muscle, they measure how fast they regenerate and how much fibrosis they get after this injury. And again, the responses were closer to the young mice. This is really astonishing. This is no drugs, nothing fancy. Literally taking the blood out, separating plasma from it, discarding the plasma, injecting saline with albumin, and mixing it with the same blood and putting it back again. And you get all of this. Isn't it great? Yes, it is. They haven't done lifespan studies yet. So I think it's only a matter of waiting to see if this extends lifespan. But definitely, even if it doesn't, it already looks quite promising. Next one, I talked earlier about atherosclerosis. So this is the accumulation of lipids and other things like macrophages, which then become foam cells. The macrophage tries to eat lipids that stick to your arteries, and then they cannot do it, so they become bloated, and other macrophages come to eat them, but they also become stuck, and they just like, the thing just keeps going. So it was thought that we cannot remove atherosclerosis other than by surgery, which is kind of nasty. But also, it seems like you have these molecules called cyclodextrins that can actually remove atherosclerosis. So these molecules are like rings of like glucose-style molecules, these like two rings. And by their structures, they act as if they were small molecular pumps that pump lipids out. So they go around this plaque and they suck up the cholesterol and the plaque. So in this case, in this one paper I'm looking at here, they used mice and they didn't reduce circulating cholesterol because one reason may be that you may be the circulation may be sequestering cholesterol from blood, but the body is still making more cholesterol, so maybe it's still being made. These are also, by the way, mice that were fed a high-fat diet prior to this treatment. And also, by the way, and in the effects of high-fat diets in humans or mice are different. So this doesn't mean that ketogenic diet is going to get you atherosclerosis. It's very specific to mice, and it's a very it's more fat than you would eat in a ketogenic diet anyway. But okay, not interesting. So they looked at a plaque area in one of the I think it was in the carotid artery of mice, and it was reduced by maybe not half, but by a substantial amount in that case. So again, this is saying, well, we can just let the lipids be. It can just because ultimately the lipids are not damaging. What damages is when the lipids become plaque. So we can just remove the plaque and problem solved. Well, but the problem so far is that cyclodextrins also cause hearing impairment, so that which is also not great. There's also work ongoing. There is a startup called Underdog Pharmaceuticals or Biotech, which is working on a better version of cyclodextrins that avoids this problem and binds to cholesterol with a much higher affinity. So this may be extremely disruptive. So currently, probably one of the most bestseller drugs that are statins, which is to reduce LDL cholesterols. And this thing may actually address the root cause of heart disease and cardiovascular disease. Hence, given that this is one of the biggest killers, if you look at mortality, it's basically cancer and atherosclerosis. This may actually go to the root cause of that, which looks also extremely promising in that regard. Again, has been done in mice. There are some trials in humans by oral administration of cyclodextrins for other reasons. Cyclodextrins are approved for other conditions, but what seems to work is to actually injecting the cyclodextrins directly into the blood so that they can actually go and scrap all this plaque. Again, we will probably see human trials for cyclodextrins sometime in the following years. We then get to combined interventions. So if you have seen all these things, you probably wonder, hey, what if we do everything together? As in, sure, if you cure cancer, but if you don't cure atherosclerosis, you're going to die from their things. So you should cure all the things. And indeed, when you do these combined therapies, when you combine the things, you usually find that the effects compound. In this case, in this particular study, they tried all these combinations. They had two drugs, lithium, rapamycin, and trametinib. And then they tried rapamycin with lithium and trametinib with lithium, and then all three. And as you can see, well, it's a bit difficult to see, but as you can see, rapamycin, which is, again, it's very robust intervention, rapamycin got them maximum lifespan of 100 years. The combination got them to 120 days in this case. And as far as mean lifespan is concerned, they also got a nice improvement. So this sounds quite like, so it's like, oh, we should give cyclodextrins and like this reprogramming thing and DNMA, we should try all these things and see how long, how far we can go. Well, the problem is that no one is doing this. I think if you look for combined interventions and on the literature, you don't really find a lot of people trying them, which may have some sociological explanations. So maybe if your lab is specialized in telomeres, you're going to study maybe only telomeres and try to see the effect of telomeres. If your lab is doing, like sirtuins, maybe you're going to study that. Because perhaps if you try like 10 things at once, it's difficult to learn about the effects. So it's difficult to do proper science, perhaps, about knowing in a very reductionist way exactly the individual mechanisms. But you're also missing out on these synergistic effects. So I think if I had like money to fund studies, I would definitely be funding combined trials to see how far we can go. And again, I'm going to also keep furthering this point with this picture that I made. So this shows an artificial case. This is not real data. This is purely an artificial case I made. So in this case, there are two diseases, so two things, two causes of death, which the mortality for these two diseases increases with age, and they're independent. So these two diseases induce, in turn, a decrease in survival. So if you have disease one, at first you have like 100 people, then you can see how many people are going to be left at each point in time. So in this case, for example, by the time In this case, only, let's say, 50% of people are going to reach the age of 80 or something. Okay, but you have both diseases present. So because you can die of either of those ways, you're actually going to, the curve is shifted to the left, meaning that people are dying earlier. And also in this case, there is also a reduced maximum lifespan. Okay, suppose you cure disease 1. In that case, you're left in the blue case where you only have disease 2. So you're increasing lifespan by a little bit, by going from the purple curve to the blue curve. Or suppose you cure disease 2, in which case you go to the red curve, which also you are also increasing lifespan, but a little bit. Now suppose you cure both diseases. Here by construction, you will live effectively forever. You will have zero mortality. So the fact that I think it is probably quite promising that even though all these interventions have maybe 10-20% improvement in lifespan, smaller effects, doesn't mean that if you put them together, you're not going to see these huge effects later on. So if you're systematic in addressing everything that goes wrong with aging, you may see these very interesting effects. Which again, say this should be done. I'm really, I think probably more radicalized about the importance of combined interventions, and it's really kind of annoying that they are not done more often. How robust is all of this? Because you may wonder, well, things don't replicate, and what if papers are fake news and stuff? Well, yes. So there are many things that have been claimed to improve lifespan. Like if you go to Laura Deming's longevity FAQ, there is a huge list of things that in some studies seem to increase lifespan. One example I want to point to here is metformin. So metformin seems to increase lifespan in mice, or it seemed to, until this thing came through. It's called the NIA, National Institute for Aging Intervention Testing Program, where they say, let's get real, let's get like thousands of mice, huge sample sizes, standardized protocols, and multiple labs doing the same test. And that test, rapamycin failed. Metformin failed. Rapamycin worked. It's very robust. So metformin failed. Similarly, this doesn't mean that it doesn't work for humans, though. This means that it doesn't work in mice. Likewise, for caloric restriction in mice, which you can see this chart on the right, there seems to be strain-dependent effects. So for some mice, so given the same amount of caloric restriction in mice, in some cases you see a decrease in lifespan, and in other cases you see an increase in lifespan, or in some cases there's no change, which may point to that, well, maybe there is an effect, but it is extremely dependent on the individual. So maybe you have to adjust the caloric restriction depending on your genes. Or perhaps because the experimenters didn't have access to the brains of the rats or to the mice, they couldn't see how hungry they were. So in humans, of course, if you overdo caloric restriction, you end up with anorexia, malnutrition, and that will reduce lifespan. So you have to know exactly how far you can go. It also matters what the control group is. So it's different if you improve aging in a mouse that's naturally aging. It's more difficult to improve aging if your mouse is like a wild type. Furthermore, it's more difficult. Like suppose you have a mouse and you're giving it like a nice diet and stuff, and that's your control group. And then how much can you do with drugs on top of that? Well, that's an interesting question that is not very commonly often addressed, and that people hope it doesn't matter much. Again, because we have, for all these drugs I've been talking about, we not only have effects, we have specific mechanisms through which they act. So in the case of cyclodextrins, you can actually take the cyclodextrins, you can put them with cholesterol, with water, and you can see exactly how they suck up and absorb the lipids. So you can see that it's not, they're supposed to be doing that, and they actually do that. So I would say that, oh yes, I also want to mention that with something about a few studies. So first, telomeres. So if you go to my longevity FAQ, you can see that telomere stuff back in the day, it was difficult to know what was going on because the assays or experiments being done to see how long telomeres were not very good. The assay they were using is called a qPCR, which for telomere length doesn't really work well. There are better ones. And in the case of mitochondria, there's also some recent debate about the assay they were using to measure if the mitochondria is producing this reactive oxygen species, that maybe that assay was also flawed, leading to, well, a whole literature built of fake news, effectively. And also, one more thing, if you read my review on immunosenescence, you can find that, well, one reason why, well, back in the day, it was thought that the immune system wouldn't attack the cancer because cancer is your own cells. The immune system is supposed to recognize your own cells and protect you, so it wouldn't attack cancer. So initially, it was difficult to prove that the immune system attacked cancer because, again, the studies were not great. The housing conditions of mice were not great, so both groups, control and regular group, would die, or if you did surgery on them, they would get infections and die. So the effect of the immune system on cancer wouldn't be easy to see. Also, they were using carcinogens, so like a highly carcinogenic chemical on mice, which people argued that induces tumors in a way that they are not natural, so the immune system is not prepared to defend against those tumors. They were also doing things like, oh, just take a tumor and you put it in a mice and the mice still dies, and it's like, sure, because the immune system can be overwhelmed by too much cancer, but that's compatible with it being able to address a small amount of cancer, around, perhaps, if you want a number, around 10 to the 5 cells. That's kind of like what a figure I've seen for how many cancer cells this immune system can deal with. And there might be, yes, there is another example of this in the epigenetic clocks literature. So the first clocks, as I mentioned earlier, didn't predict mortality very well. And the reason for this is that they were trained on longitudinal samples. So these are samples that you take a bunch of people from different ages, you sample everyone once, and you train your data on that. The problem with that is that if you are a fast ager, I think if you are epigenetically older, you will die sooner, right? So you will not be in the sample. So when you're sampling older people, you're sampling people that have survived. So those data sets were introducing a survivorship bias into it. Now the second generation clocks, those used cohorts. So they used the same people sampled multiple times over their lifetime to reduce this bias. And that way they were able to pick up those CPG sites, these methylation sites, that actually correlate with accelerated aging and were able to show a huge effect. In fact, these epigenetic clocks, the second generation ones, they are as predictive as smoking or not smoking. So suppose that you want to predict someone's going to die, and you can tell me either if they smoke, I think it was like 12 cigarettes or none per day, or if they are in the highest or lowest deciles of this epigenetic risk score. It's better to know the epigenetic score than if they're smoking or not, to see what the mortality risk is. Okay, so we are getting near to the close of the presentation. Now we have only a few slides left. So this one is about, well, the FDA, that they don't consider aging to be a disease. This is very subjective. So what is a disease? That's just like, it's more of a role for, it's purely a semantic debate. But as I say, definitions matter on a case-by-case basis. And in this case, it matters because the FDA cares if something is a disease or not. Now, making aging into a disease is complicated because what is aging? It seems to be many things. So the FDA has been reluctant to classify it as aging. This is not all lost, though, because you can still approve these drugs for a single indication. So, for example, rapamycin is not approved for longevity. It's approved as an immunosuppressor and some rare disease. But some doctors can prescribe it for longevity if they want. So in the US, at least, doctors can prescribe so-called off-label any drug for anything they want. So it's not too bad. So say likewise, Unity Therapeutics, Unity Bio, it's despite the fact that senolytics may work for everything, they're still targeting individual things. They're going after arthritis in particular, and then they can say, well, this thing also works for something else. Lo and behold. However, this is an issue for sites because it makes them more expensive. So suppose that you want to do a drug that prevents heart disease. Now, if you enroll a cohort to prevent heart disease, because these events are rare, they take years to unfold. You need to have enough statistical power to prove that your drug works. You need to have a lot of people and wait a long time, and that costs money. And if, for example, if someone dies of cancer, that wouldn't count for your drug. While if your drug is an anti-aging drug that reduces cancer odds, you wouldn't be measuring cancer. So in that sense, anti-aging drugs in the current FDA paradigm, the studies you would be using are underpowered to detect the effects in aging. So in that case, even though the drugs can get approved anyway right now, recognizing aging as a disease can make testing anti-aging drugs cheaper. So we may see more of them. And so in that regard, there is this trial it's called targeting aging with metformin, and the idea here is to use metformin because there's some evidence from some trials that in diabetics, non-diabetics, diabetics that take metformin seem to live longer than non-diabetics, which is kind of interesting. And this might be perhaps with a it also goes to AMPK, which is also is tied to mTOR, and also because it's reducing blood sugar, which we have seen it's also not great for you. So what they did here is this was led by this guy called Nir Barzilai, and what they did here is to agree with the FDA on a composite endpoint. They said that they will count as a thing they want to prevent as a composite of any of time to new diagnosis of cardiovascular disease, cancer, dementia, or death. So this is what they are measuring. So any of these events is an event that they want to prevent, which it seems it's a reasonable proxy for aging. You're kind of like bagging together into one place the key mechanisms, and we saw earlier in the first slides, it's CVD, cancer, and dementia, or just death directly, that are driving mortality risk. I said so. Metformin was not so great in mice. It seems more promising in humans, and so I think this trial is quite welcome because even if the effect is small, even if we're finding a small effect, we will get aging. The FDA promised that if it succeeds, then other studies can use the same endpoint for their studies. So we will have an aging endpoint finally, to sort of other drugs can be tested and validated and approved using this endpoint. Okay, so I've mentioned some controversies already, some things people disagree about in the field, some open questions like you could, for example, how much should you focus on reducing damage or aging production of damage versus removing it? So the Sense Foundation, they are all about let's damage happen and let's just remove it. Fixing metabolism is difficult. Most of the field, I think it's more on actually we can go and fix things. Why not both, of course, but that's a question. Second one is how far can we go? As in, we have shown that in mice, well, we have shown that in worms we can make them live 10 times longer. In humans, we know that there's lots of variation in age. In mice, the most, I think the largest increase might be 40% maximum lifespan, perhaps. I don't know. And this was with genetically engineered mice. It's like dwarf mice. So again, how many years we can add? We don't know. I mean, we don't have a proof that says we cannot live more than this age, but we don't know yet. Third one is once we have aging reversal, what does that mean in the first place? So if you give someone statins and reduce cholesterol, sure, they will have their mortality decrease. Does it count as an aging reversal? Maybe should we just look at mortality? Should we have epigenetic clocks or maybe multiple epigenetic clocks looking at different organs and how they age? Or should they have more of a proteomics looking at proteins, at protein levels in the whole body to measure their levels and see if they're healthy levels or not? So we can actually have, or should even probably have health scores per organ, because probably each organ ages individually and in a different way. Next one is, what are the main blockers in the field? Why is not the field advancing faster than it is? One may wonder. This kind of question to think about. One of them, I think, is the one about combined interventions, is that they are not done enough, and this may be because each lab has its own field, and combined interventions require more cooperation and across labs. But something to think about. Fifth one is, if right now, without any new discoveries, if you just combine all those things, how much lifespan would you get? Or how much of an effect would you have? That has not been tried, and I think should be tried to see what is already on the table without any new discovery. And sixth one, how do the hallmarks relate to each other? So we have seen that kind of the immune system is connected to the brain and to metabolism and inflammation. Everything seems kind of tied together. But this is not necessarily very useful. We want to know exactly how they are tied together so we can devise better interventions. And right now, this is not that well known. To finish off this thing, 7 points. First one is that, well, lots of unknowns. We don't know what everything not aging yet. Second one is that the knowledge in the field is pretty much in flux. Loads of things, and you have seen many of these papers I've been citing are really recent, and just from either this year or the previous year, and it remains in flux, which is very exciting. It means that it's it's not an stagnating field. It's lots of progress in there. Third one is that we know now that the process of compressed aging can be slowed down and reversed across species. Fourth is that these processes are not separate; they are interconnected. So improving one process can potentially improve others, which is also nice. Fifth one is that progress in this field seems to be accelerating. So we are seeing more and more startups, more and more discoveries, and more funding going into this area, which is also nice. Sixth one is that these therapies are not hypothetical. They are already in the clinics. Some of them are already in trial. And seven, I really want to hammer this. We need more combined therapy trials. We need to see exactly, we need to tackle all of this, because maybe we have fantastic cures already available, but we don't know it because we're not testing them, and we definitely should. Yes, yeah, I wanted to acknowledge Adam Marblestone and Laura Deming. They have helped me with, they gave me feedback for the slides, and I didn't take any money for doing these slides. I just did them in my spare time, just taking all the knowledge that I have accumulated over maybe the last year or so, which I've been reading about this topic. So thanks for watching, and if you want to know more, as I said, you can go to my blog to read about this stuff. You can subscribe to my blog. I have a Patreon if you want to give me free money. That would be nice. And that's pretty much it. There are a few topics I have not discussed because the field is, I mean, you could go for days talking about these things, but I just wanted to compress which I think are the key things in the field. And while making these slides, I was trying to look at other presentations from other people and to try to kind of convey so much knowledge in such a way that I think currently there shouldn't be any better presentation than this one available. So I think that you pretty much have a view of the state of the art in the field. You have a view of the controversies that have arisen. You have pointers to where you can read more about the field if you want to learn more. And that's pretty much it. So thank you for watching, and goodbye.